All right, let's get started. Hi, everyone. Welcome, and thank you for attending the first session of Vector Builder's new Gene Delivery Seminar Series. Our speaker today is Dr. Priyanka Narayan, an NIH Stedman investigator and the acting section chief of the Human Diseases section of the Genetics and Biochemistry branch at the NIH NIDBK. After completing her PhD at the University of Cambridge, Priyanka worked as a postdoctoral fellow at the Whitehead Institute at MIT before starting her own lab at the NIH. There, her lab uses biochemical, genetic, and neurophysiological studies in induced pluripotent stem cells with the aim of identifying novel therapeutic or preventative mechanisms for neurodegenerative diseases. So following the presentation today, we're going to have a Q&A session. So please type any questions you have into the Q&A text box, and I'll read them out at the end. And now I will hand things over to Priyanka.
Fantastic. All right. Well, thank you so much for that introduction, Connie, and thank you all for being here. It's a pleasure to spend the next, 40 minutes or so telling you a little bit about my experience in the field of using induced pluripotent stem cell models to really capture the cell biology and the fundamental molecular underpinnings of neurodegenerative disease risk. So I'll start by kind of talking a little bit about the problem that we're trying to address, how iPSCs have been really enabling in this field and really have advanced the way we can study human cell biology, especially in brain cell types. And then I will kind of highlight some of the opportunities that gene delivery has really opened up to be able to manipulate these systems in ways that we haven't been able to. So just to introduce the disease space that our lab and many others are very focused on, and undoubtedly many of you have heard of this, Alzheimer's disease. It's the most common neurodegenerative disease on the planet right now. Its incidence is rising exquisitely with the increased age of our population, and it's characterized by a severe loss of brain tissue, which you can see kind of exemplified by this very striking images. What those of you might not be as familiar with is that only 5% of Alzheimer's disease has strong autosomal dominant inheritance patterns. So these are patients that usually suffer from symptoms before the age of 65. And the 95% of this pie is actually characterized as late onset Alzheimer's disease or sporadic disease. This is the more common version of the disease, and it has no causal genetic factors. But I think that raises the question, given how common the disease is, why do some people get late onset Alzheimer's disease and others do not? And with the advent of genomic sequencing, this has been something that we've been able to try to address, at least on the genomic side of things. So what technology has really enabled here is genome sequencing studies of large swaths of the population. These are people with Alzheimer's disease and age-matched controls. And they've sequenced their genomes and found SNPs, so single nucleotide polymorphisms, changes in their DNA that are peppered across the genome that associate more highly with Alzheimer's disease patients than age-matched controls. And you can see some of this data presented here in a Manhattan plot, so you can understand why it's called that. Each one of these dots, these are the chromosomes laid out on the x-axis, and each one of these dots represents a single polymorphism in the genome that's enriched in the population with Alzheimer's disease relative to the controls. So as kind of a cell biologist, a molecular biologist on the receiving end of such data, what I really want to know, and I think many others as well, is what do each of these polymorphisms, these DNA changes do? So I think, we try to figure that out. And there's a real question of how we try to go about this process. What are these risk polymorphisms doing to cells? How does cell biology change? What does risk mean on a molecular level? So this has, in the cell biology field, conventionally been undertaken using cancer cell models for other diseases. These are HECT cell models, HeLa cell models, and many others that I'm sure many on this call are familiar with. But these cells, although they have been workhorses of cell biology and offered many insights into really fundamental discoveries, they lack a lot of the really specialized biology of brain cell types. Brain cell types often have different structural elements. They have different functional elements that may not be captured in these more generic, fast-growing, dividing cancer cells. So really, we would like to study human brain biology in human brain cells. And as you can imagine, getting human brain cells is not an easy feat. No one is volunteering biopsies of healthy patient cells, and primary cells are extremely difficult to obtain. But, and I say here, they're hard to obtain until recently. That gives my scale of recently. But in 2012, and thereabouts, this has changed a lot. And this was awarded the Nobel Prize in 2012, this discovery by Shinya Yamanaka and colleagues, but that they could take somatic cells, in this case a fibroblast, but this can be applied to other somatic cells as well, blood cells as well, and they can apply 4 transcription factors. Another example of how gene delivery has really revolutionized a field. Apply 4 transcription factors to these somatic cells and create what's called an induced pluripotent stem cell. And this has been really remarkable because now you can create a stem cell from any human. And stem cells, as many of you might be aware, these iPS cells, amongst other types of stem cells, can be directed through differentiation pathways into different terminally differentiated cells with different, what we call cell fates. So these terminally differentiated cells are replicas in a lot of ways of the cells that exist in our body. They have slightly different biology, but overall they are far better replicas than, say, a hex cell or a HeLa cell of a neuron. So this technology of being able to induce pluripotency in any cell, any somatic cell, and be able to derive that into a terminal differentiated cell has really enabled us to be able to study specialized brain tissue biology. And iPS cell protocols to differentiate these cells have been developed over the years by many, many groups. And they have been applied to a number of cell types in the brain. And I'm showing you just a few of the different cell types here that are integral to brain physiology. There are vascular cell types like endothelial cells and parasites that form the vasculature of the blood-brain barrier, which is supposed to be extremely important in brain function, of course, and also in disease. There are oligodendrocytes, which are involved in the myelination of axons, the sheathing of axons of neurons to allow communication. There are glial cells, which I'll talk about a little more later, astrocytes and microglia. These are traditionally thought to be immune or metabolic support cells for a number of other cell types, but now they've actually gaining appreciation that they have their own roles in cell-to-cell communication and transmission of electrical signals as well. And there are, of course, neurons, which I think are the ones that degenerate in the end and the ones that neurodegenerative diseases really would like to preserve. So there's a lot of biology that we now have access to due to these cellular systems. Now, these are cartoons from BioRender, but what do they look like? So I'm just going to give you a few examples of, in this case, it's a co-culture of astrocytes and neurons, and you can see the really beautiful physiology and how different these cells look than the HEC and HeLa cells that we had in culture, and how these cells can, as you can imagine, just by looking at their structure, capture really different biology, motility dynamics than our traditional cell models. Now, I wanted to spend a second talking a little bit about how we get iPSCs to these terminal differentiated cell fates. There are multiple different protocols, as I suggested before, and these kind of fall into 2 larger families. There are small molecule differentiation protocols, and these tend to follow or try to recapitulate the natural differentiation pathways that iPS cells undergo to differentiate, or stem cells undergo to differentiate to terminally differentiated cells during development. So these involve addition of various factors that are the growth factors or patterning factors that can direct stem cells down certain lineages that, and then kind of narrow that search space of their differentiation. Then that is a more conventional way of generating these terminally differentiated cell types, and it's been extremely effective for years. However, there's kind of a new wave of cell type generation that has been emerging over the years, and this is called a transcription factor mediated differentiation. So what these differentiation protocols usually do is take an iPS cell and using kind of a retrograde analysis of what is expressed in a downstream cell very highly, they take transcription factors and overexpress them in iPS cells to really drive these iPS cells, these stem cells, to a certain fate, for example, a neuron. These do not necessarily recapitulate the developmental process that a stem cell would normally undergo in a developmental, during development to achieve a neuronal fate, but the final end stage cell type looks a lot like what it would look like if you had undergone that process. And, the looks a lot morphologically, transcriptionally, et cetera, et cetera. So this is the real advantages of this approach are you get far more homogeneous cultures. These can be adapted to larger scale experiments, such as screening and other types of large scale techniques that require a lot of cells to do them. But if you are studying how a cell develops from a stem cell to a terminally differentiated cell, there is a disadvantage there because you don't recapitulate that process phase. So there are advantages and disadvantages, but given that a lot of iPS cell biologists that are studying neurodegeneration really want to study these end-stage cell types, this has been quite an enabling technique in this field. And in fact, a lot of the, just to kind of think about gene delivery in this context, a lot of these transcription factor mediated protocols have been enabled by efficient lentiviral delivery. So integration of these transcription factors in inducible cassettes into iPS cells so that you can direct them to neuronal cell fates. Or another kind of slightly different integrating approach is using piggyback transposons approaches to integrate various transcription factors into the genome of iPS cells and inducing them using, say, a doxycycline. So this is another way that gene delivery has been applied in this context to really enable the study of these more complicated cell types. So now kind of getting back to the problem that we were really trying to model or get to the molecular independence is how do we model mutations in disease risk genes? So as I mentioned to you, we can take skin samples from either patients or healthy controls. We can use gene delivery to induce pluripotency. These are usually episomal deliveries so that you don't have integrated constitutively iPS cells. And then you can integrate transcription factors or use small molecule approaches to derive terminal cell fates like neurons and the other cell fates I discussed. These final neurons can be used to apply cell biology techniques to look at disease processes that might be disrupting fundamental cell biology. You can use it to test drug candidates, to understand genetic interactions, and really to kind of use that information collectively to develop therapies. But I mentioned to you that, a lot of these associations, these risk associations from the genomics that have come are not causal genes. Only 5% of Alzheimer's really is autosomal dominantly inherited causal genes. The rest, there are many people with those genes that do get disease, but there are also others that don't. So clearly they're interacting factors, both genetic and environmental, that might affect that. So how do we really capture that heterogeneity when we're modeling disease risk? So there are kind of two different approaches that have been taken in the field to do that. The first is to up our N, right? So if we want to see the difference between cases and controls and kind of try to distinguish signal from noise, we really have to have a lot of cases and a lot of controls. So this involves comparing a large number of control lines and disease lines. And depending on the effect sizes of the phenotype you're measuring, that might be a very large number, might be a slightly smaller number, but this can get, it can be manageable, but it can also get unwieldy depending on how small an effect you're measuring. So this has been one approach that has proven effective, but is best applied to very, very large, strong phenotypes. Another approach that has been used to kind of pinpoint the effect of single point mutations in isolation of effects of genetic background is to use isogenic cells. So this is kind of what I'm lumping in this context of creating control and risk versions of the same person's cells in a dish. So ideally, we would want to take a person, whether they're a control, for example, if they're a control, take those cells, induce the Alzheimer's risk mutation that we are interested in studying, and then compare what their cells look like in the risk case, genotype case, and in the control genotype case. And really there you have a perfect comparison. And maybe do this across multiple sets, but really have really strong controls so that the deltas that we see, we know are attributed to those mutations only and not other gene interactions. So this is something that has also been enabled by gene delivery. This has been done most recently and kind of most commonly by CRISPR-Cas9-based gene editing, delivery of guide RNAs to iPS cells, either through ribonuclear protein complexes or other mechanisms, and then using CRISPR to cut and repair and create mutations and create isogenic cells. So this is an approach that we definitely use in the lab. We also try to profile different sets of lines, but I think this has been a really enabling technology where gene delivery and gene editing specifically has enabled the study, really highly controlled study of point mutants and their effect on cell physiology. So with kind of that background, I wanted to showcase a project that we're undertaking in the lab that is using these approaches to understand really fundamental cell biology mechanisms that underlie Alzheimer's disease. And I think you'll see mentions of all the various techniques and how we've applied them in one circumstance and how various, and this is a project that I think has been really enabled by our purchases from Vector Builder. And I'll show you how we've been able to use lentiviral constructs to not only develop these cells, but also modify them and study genetics in a new way. So this project is about endocytosis. So it's about how cells take up cargo and extracellular substrates from the extracellular environment and internalize them. And this process, as you can imagine, many of neuronal communication processes and glial communication is based heavily on this process. So it's a central process to many brain diseases, brain function, and is perturbed in multiple brain diseases. So being able to understand how this process is modulated in the context of Alzheimer's disease is, I think, a very central part of understanding the molecular biology of pathogenesis. So let me just introduce you to endocytosis for those of you who might not think about this all the time. So clathrin-mediated endocytosis, which is something that I'll talk a lot about today, is one mechanism by which cells uptake material from their surroundings. So various substrates, various substrates are taken up from the extracellular environment, here kind of depicted in this cartoon, through these vesicles. And these vesicles are formed and kind of caged in this scaffold of molecules, which include clathrin, a structural molecule for this, but also various adapter proteins, which I'll talk about in a little bit, which structure how material is taken up. And these are captured into early endosomes. Then those are trafficked down the kind of endolysosomal pathway to late endosomes, to lysosomes, can be redirected to recycling endosomes. And each of these various compartments, part of the endosomal pathway, are marked by certain proteins that come on, dock on the vesicles, and leave based on what stage of this pathway. And so really timing and marker proteins. So this is a pathway that we know has been perturbed in the context of Alzheimer's disease. And we know this not only from just understanding normal neuronal and glial physiology, but really a hallmark, I mean, this is a paper from over 20 years ago, but a hallmark of AD pathology is accumulation or stalling or disruption of endosomes in patient tissue. And this has been seen Alzheimer's and Down syndrome in a number of neurodegenerative diseases. And more recent work that I'm actually not showing you here has even shown that inducing defects to early clathrin-mediated endocytosis can induce neurodegenerative disease defects. So clearly the two, neurodegeneration and endosomal trafficking, are intimately linked. Moreover, I think we know a number of risk factors, and I'll point to a few more of these, but we wanted to kind of understand how these risk factors play into endosomal trafficking. And our lab, one of the risk factors we spend a lot of time thinking about is this gene ApoE. So ApoE is a secreted protein. It is a modulator of lipid biology, but we wanted to see how this extremely, and it is the strongest risk factor for Alzheimer's disease. It comes in various flavors. The most common variant that most people have is called ApoE3. About 80% of the population has ApoE3 as their allele. And this is considered neutral, or it's kind of our reference allele for Alzheimer's disease risk. Whereas a single point mutant, a single coding point mutant, in this case, a cysteine to arginine mutation that results in an amino acid mutation, results in the strongest AD risk factor in the context of sporadic disease. Not Mendelian, but it's still the strongest risk factor, and it is highly penetrant for risk. So we wanted to kind of understand the intersection between these two parts of biology, how risk intersects with endosomal trafficking. So we decided to look at how ApoE isoforms E3 and E4, the neutral and the risk variant, impact endosomal trafficking. And do we actually see differences between these when we look on a cellular level? So I'll share with you some of the data, but I wanted to introduce the system, which is kind of a callback to the work that I talked a little bit, the approaches we talked to earlier. So for this work, we used multiple sets of iPSC derived, in this case, I'll show you data from astrocytes. So we took cells, these are fibroblast generated iPSCs derived from first a late onset disease patient with an E4. So you can have two copies of the ApoE allele. So it's E4 homozygous, so a risk allele patient, or a healthy individual, a different person with an E3 genotype. This is a neutral non-risk genotype. And of course, using CRISPR-Cas9 genome editing, we can generate risk and benign versions of that person's cells with respect to their ApoE alleles. So we have two isogenic sets of E3 and E4 alleles. Here again, an E3, which is we call the parental line, and then E4, which is the edited line. And we've done all the experiments that I'll show you in both contexts, just to confirm independence from other genetic backgrounds, factors that may be influenced. So just to kind of show you what this looks like in the lab when we stain our iPS cells, they don't look as ramified and beautiful as astrocytes in a brain, but they do have hallmarks. They stain for the conventional markers of astrocytes. Some of them have kind of more processes, but these are cell types that are replicas, I would say, of brain cell types that we can really use to study the cell biology in. So we've generated astrocytes from these isogenic lines, and we decided to just see how endocytosis is perturbed. Now, you might be asking, why am I looking at astrocytes and not neurons? Neurons are involved in neurodegenerative disease. Neurons use endocytosis. Well, we started looking at astrocytes because we're studying ApoE. And for those of you who are unaware, ApoE is most highly expressed in astrocytes and actually quite lowly expressed in other cell types, including neurons. So we wanted to make sure that if we're studying an ApoE-dependent effect, we look in cell types that ApoE is most relevant. So we decided to look in astrocytes, which are integral to synaptic communication and regularly exchange metabolites using endo and exo, or substrates using endo. So I apologize for the slight graininess in this slide, but the result I think is very clear. You can see that when we compare ApoE3 cells and ApoE4 cells, here staining for an early endosomal marker in these little white dots, you can see that our ApoE4 astrocytes, each of these little cell areas is an astrocyte. You can see that there is a markedly decreased level of endosomal staining in the E4 astrocytes than in E3. So this kind of gave us that our E4 cells are deficient in endocytes. Something about the ApoE4 genotype confers a deficiency. We can also look at the active process of uptake. That was the static picture saying that, you know, at a certain time point, constitutively ApoE4 cells have lower endocytosis or endosomes, but maybe we can look at how well they uptake new substrates. And we can do this using various ligands. So we can use fluorescently tagged ligands like transferrin, which targets to the transferrin receptor, or EGF, which targets to EGFR, both of which are expressed on multiple cell types, including astrocytes. These are very traditional assays to probe endocytosis. And we can look at the flux of these ligands into the cell over short periods of time, which really focus on that early endocytic abnormality. So we do this, we pulse the cells with ligands, and then we look at right after 5 minutes, how much they've taken up. And we can see that here on the left in transferrin and on the right in EGF, the ApoE4 cells take up way less of these ligands than the ApoE3 cells. So we can see that there's clearly a deficiency, similar to what we observed with endosomal staining, in the uptake of ligands. Now, this is nice. It's nice to observe something, but it's always it's even nicer when you can disrupt the system and try to understand how it works and maybe even alleviate defects that might underlie disease. So we asked the question, can we actually rescue these disruptions? And for that, we actually turned to a number of sources of inspiration for how to do this, one of which was these genomic studies again. A number of other factors, and I'm not going to point all of them out, but I'll mention a few: BIN1, INPP5D, but most notably PCOM, are other genes in which polymorphisms have been identified, or proximally to which polymorphisms have been identified, that are also associated with Alzheimer's disease. So we thought the genomics is pointing to endosomal disruptions, but maybe these pathways are not distinct. Maybe they can actually interact. The genetic associations with risk may not occur independently with each other. And this was something we also could corroborate with more literature, where we saw SNPs in this gene PCOM, an early endocytic adapter protein, were associated with Alzheimer's disease, and these SNPs decreased expression. So we thought, okay, if less of these endocytic protein adapter SNPs increase Alzheimer's disease risk, can we bolster this and maybe bolster PCOM expression and then alleviate the defects imposed by this other risk, ApoE? So can we actually look at gene-gene interactions in the context of risk factors? So we did just this, and this is where, and I just want to tell you a little bit about where PCOM lies in this pathway. I mentioned to you clathrin forms this cage, and one of these adapter proteins that really enables the cage formation and the linking of different substrates to that cage formation is PCOM. So it lies really integrally. It's a structural protein in that process. And so we decided to use some gene delivery methods, which I think have been really enabled by technologies, lentiviral delivery, where we can actually genetically modulate these cell types. And these cell types, iPS cell types, have had mixed effects with how strongly you can genetically modify them. Different cell types need different elements to genetically target them. Cell type specific promoters like GFAP in the context of astrocytes are really helpful, especially when you're doing experiments with multiple cell types in a single dish. And then different elements to enable stable expression in the constructs of these lentiviruses are really important. So these are things we've started to explore and play with. So we've used lentiviruses, either control lentiviruses or lentiviruses that express these early endocytic adapter proteins like PCOM to target these astrocytes. We took ApoE3 and ApoE4 astrocytes, our benign and our risk astrocytes. We tried to bolster them by overexpressing this early endocytic adapter protein to see if we can promote or correct a deficiency in endocytosis. And what we found, so we applied our assays, the fluorescent uptake of ligand assays, so we can look at how quickly they uptake ligand. And we saw, and I'm just giving you one example of the data, this is published data, so you're more than welcome to find all the rest in the publication. But we can see that even with just delivery of a single risk factor or a single gene here, PCOM, we can increase the uptake. And it's pretty obvious when you look at it by eye, and you can see a strong effect quantified here. So you can increase the uptake of various ligands into the cell. And one just kind of fun biological point is this is not, so that this is ApoE4 data. So cells that are already deficient in endocytosis can be rescued with the overexpression of PCOM. But this is not true for cells that are competent in endocytosis, like ApoE3 cells. If you overexpress PCOM, they actually are more deficient in endocytosis. I don't have the data in this presentation to share that with you there, but it is in the publication. So there are clearly gene isoform or gene variant interactions that are specific, and they target the cell biology that's impacted by these various genetic factors. So now I just wanted to share some of the directions that we're taking this similar types of work in the lab now. We tried one risk factor here. We tried PCOM, and we found that it worked, which was great, but it is not structurally that different from many other risk factors. There are other adapter proteins that either function as risk factors or that at least function in the clathrin-native endocytosis pathway. So we want to know, how specific is this effect? Is PCOM the only protein that can affect ApoE4 induced defects? Can other early endocytic adapters that have similar function or similar structural elements to PCOM do the same thing? And for this, a graduate student in the lab, Artur Gevorgian, is trying out a number of these candidates using a very similar approach, again, using lentiviral gene delivery methods that we've partnered with Vector Builder to really be able to try out and exploit in this context. So we use ApoE3 and ApoE4 astrocytes. We deliver modifiers using these, and these are candidate modifiers that have similar associations with risk or similar functions as PCOM. And then we apply these cell biology assays. And we can, and I just wanted to, because this is in the context of gene delivery, share kind of some of the plasmid design elements. I know a lot of people probably think about this more, but we can change promoters to generic promoters that might be applicable to more than one cell type, to cell type specific promoters using elements like GFAP to target astrocytes or synapsin to target neurons. These have selection markers for both fluorescent selection and antibiotic selection, and stability elements like WPRE. So we're really kind of exploiting the toolkit and stuffing these lentiviral vectors pretty full to try to get as much functionality out of these experiments as possible. And just to show you, we don't have all the data on this yet, but just to show you some of what this looks like in practice, I have kind of a zoom in of a field where we have a transfected cell amongst others, untransfected cells. And you can see here, just with one of these candidate modifiers, you can see the punctate localization, suggesting that these gene delivery methods are delivering functional proteins that we can visualize using tags like HA, even if their endogenous antibodies aren't great, and we can see that they are localizing to puncta that we would at least appear to be like endosomes. So we're really able to manipulate these systems in cell types that I think were previously intractable, but due to a number of advances in technology over the last couple of decades, we've really been able to study deep cell biology in highly specialized cell types. So just to summarize what I've covered with you today, I think I've tried to demonstrate to you how enabling iPSC-derived cell types have been to really get at the molecular and cell biology that underlies brain disease, but also neurodegeneration in our case. Gene delivery has really enabled both the field of transcription factor mediated differentiation, so we can get from an iPS cell to a neuron or a neuron in a dish in, say, 28 days, astrocytes similar. And these cells do faithfully replicate many of the aspects of physiology of brain tissue, the endogenous brain tissue. And then gene delivery, I've shown you in our last example on endocytosis, and we have many others that really enable the genetic manipulation of these highly specialized cell types in cell type specific manners with promoters and different design elements of these terminal differentiated cell types. And we can really use these as a toolkit to probe the cell biology of neurodegeneration. So with that, I will just acknowledge our lab, which has been a pleasure to have. And also to Connie and Sophia, who we work with a lot at Vector Builder, and also the funding sources that support our work. And thank you for your time and attention. I'm happy to take any questions.
Awesome. Thank you so much. That was a really fantastic presentation. So we have a few questions already. Please type your questions as they come through into the Q&A. So the first question, have you tried proximity labeling techniques to identify the other potential APOE for interacting proteins?
It's a great question. We would love to do this. It is a ER targeted protein, so we are working on ways to enable that. So it's a great question. We have not fully tried it. I cannot give you answers to what the data will look like, but I think that's a great idea. I agree. And hopefully we'll have some data to report on that at some point.
Perfect. And then the next question, whether the endocytic defects in astrocytes have any connections with other pathological features like plaques or tangles? That's the first part of the question.
Yeah, it's a great question. So one of the primary thoughts around endocytosis and plaques is that glial cells like astrocytes and microglia are responsible for breaking down, maybe not necessarily the plaques themselves, but the precursors or uptaking and recycling precursors, prefibrillar species like amyloid oligomers. So this has been one of the, I think, main conventionally thought pathways in the field for clearance of oligomers. So you could imagine that deficient endocytosis would lead to deficient plaque clearance, or at least more amyloid precursor or more amyloidogenic precursors that could lead to plaque accumulation. Now, we have not shown, so there have been multiple studies towards this that are demonstrating this in a number of contexts, in the context of endothelial cells, astrocytes, microglia. So definitely there is a consensus around that is a major pathway. But in a mouse model or in a human model to really see that deficient endocytosis is leading, we haven't done those manipulations, obviously, but I think it's something that we know that they're correlated very highly in the more complex model systems, and we see the evidence on a cell biology level. So those both point to that.
Right. That makes sense that, the sort of genetic entanglement is going to lead to a morphological entanglement, essentially.
For sure.
So the second part of the question is, why use lentivirus and why not use AAV?
Yeah, great question. So a couple of reasons. So with our astrocytes in the lab, we can actually keep them in culture quite a bit, and they do divide. So we would prefer to not have an episomal type of system, a system. We prefer an integrating system so that if we need to expand them, at least for a passage or two, or do larger scale studies than our cell biology assays, that we have the ability to control that the cells in our population, or we can select for cells that actively have the integration of the desired modifier. AAVs have been used a lot in the context of the mouse model biology, at least to study perturbations. Lentiviruses, and this is, I think, Connie mentioned when we were thinking about this seminar series, issues that have not really been talked about in the field, but I generally see people have had higher success in the context of iPSC genetic modification with lentiviruses than with AAVs. And people have tried. Why that is, I'm not sure. Because I know in human brain systems, mouse brain systems, and other organisms, AAVs are a stalwart and they're, a tool of choice. That has not really been the case in the iPSC. So that might be for unknown genetic elements that have yet to be really well understood and discovered. But yeah, for two reasons, we choose that.
That's really interesting. And yeah, very clear answer. Thank you. So we have another question that's regarding differentiating iPSCs. It's kind of a similar question, so I might modify it slightly to say the question is asking about using mRNA for iPSC differentiation, but I'm gonna, since you kind of already answered that, I might flip it around and say for your CRISPR editing, are you using lentivirus to deliver those CRISPR editing components, or are you using something like mRNA for kind of more scar-free approach?
So just for the differentiation part of it, just one note also to add to the previous thing is these differentiations are not short, right? They are like a month on average of scale. Maybe the fastest protocols with transcription factors can at least be 2 to 3 weeks. So, and then cells are dividing, especially iPSCs are dividing. So we need to have integrating methods for those. Because we don't necessarily keep any selection on the cell type during the differentiation process. So we want to drive home as much of that transcription factor for as long as humanly possible to get the cell type that we want. But for the CRISPR editing, it's actually the opposite consideration. We want it to get in there, do it what it does, and get out so it doesn't do too much. And we want to be able to select for the ones that we want. So we have the capability to select for the ones we want. So it is kind of different considerations how long you want the gene delivery persistence to be. So for the CRISPR editing, we generally do it with RNP-based, nucleofection-based protocols rather than using integrating viruses. For anything that we want in there forever, as long as humanly possible, we use our lentiviral protocols. For other ones, we don't.
Perfect. Excellent. And then our next question is, how can we target gene editing, for example, in astrocytes?
Target gene. So
I'm guessing that means kind of in vivo. Like, can you in an in vivo system, can you target these gene editing components to astrocytes?
So if you are doing things like AAV-based expression, right, there are serotype preferences. I know on the Vector Builder website, there's a great table of all the serotypes and all the different cell types they seem to infect. And I think in practice, there are slight differences from model system to model system, but there are serotype-based preferences on different tissue types that are preferred for different gene delivery systems. But I think there are also the toolkit of promoter choice. So promoters, cell type specific promoters are extremely powerful. The thing with, we just have to make sure that these things are, especially for gene editing, that if you want it on forever, it's there forever. And if you don't want it on forever, you can get rid of it quickly, right? So it's kind of just making that decision call, and especially in the context of therapeutic delivery, how, when, how often, how much. These are all really important questions to think about in the context of gene delivery or gene delivery. But in terms of editing, AAV-based methods, I think, have been one of the more common methods to deliver editors to brain. So.
Perfect. That makes sense. So our next question, can primary astrocytes be transfected with transposons for genomic integration? And if so, how would the efficiency compare?
Great question. I have not done that experiment, so I do not have a quantitative answer for you. That being said, I know that in culture, iPSC-based astrocytes have been targeted with piggyback, the terminally differentiated cells have been targeted with piggyback-based transposon methods. Those are effective, but in the few examples that I've seen, I think lentiviral delivery seems to be more effective. Of course, everything can be optimized based on, lipid carrier and the various DNA elements that you need. The carrier elements and the kind of the mix that you need. So I can't definitively say that it is by far worse. I don't think I have a good answer for that, but it can work. So I think just the efficiency probably needs to be optimized. But I don't think the majority of gene editing in the context of iPSC work in the piggyback realm I've seen in the iPSCs themselves, and that is used routinely. They're very easy to transfect with piggyback transposons, piggyback vectors.
Perfect. That's great to know that there are some easy to transfect cells.
Yeah, for sure.
Excellent. Our next question is, do you have any general tips for lenti transduction in iPSCs and selecting or creating clonal iPSC lines?
That is a fantastic question. This is something actually that comes up in the iPSC field a lot with clonality, and especially with gene editing processes, but also for lenti integrations. So generally, when we have used gene delivery to, for example, integrate NGN2, a transcription factor that's used to drive neuronal lineages, into iPSCs or other transcription factors, we haven't used clonal selection. The reason for which is that I don't think we know the ideal gene integration number that yields the best neuron or the best astrocyte, right? So having a population in some way is safer because we don't really know what to select for, because we might have too much, too little, and everything in between, and we actually don't know which one will work. Now, if someone would like to go do that experiment, please, it will be a great experiment. I'm sure everyone in the iPSC field would rejoice. But really, I think it's one of those things that the bulk of people that use lentiviral piggyback induction to do transcription factor mediated differentiation will usually not use clonality or will not use clonal lines. They will transfect as a pool and differentiate that entire pool out. Now, that might mean that you get variability, right? You might get some with 5 copies and one with 1, and who knows what variability you get. You can minimize that a little bit if you do a counter selection, say on fluorescence intensity of a co-expressed fluorescent protein. You might be able to control for integration number a little bit. So you get, the highest expressors or just the lowest expressors. But I think a lot of people have used this kind of bet hedging approach where you just go with the pool, because as long as they contain at least one and they're selected for at least a single integration, then you'll get something. And we don't really know what the goalpost is in terms of the number of integrations. That being said, for other types of integration, and we use, again, just to clarify for our terminally differentiated cell types, we also use that similar approach. Our manipulation is not clonally selected, right? We are using whatever gets integrated, we profile, but a lot of our readouts are microscopy based, so we can look cell to cell and say, this has a higher level of expression of the target protein. Does it have a higher level of whatever phenotype? So those approaches allow you to at least control for that a little bit.
That makes a lot of sense. And then carrying on from that, due to the insertional mutagenesis of lentiviruses, will terminal differentiated astrocytes that are coming from one single sort of progenitor cell, are they going to have the same genetic profile? And is that kind of taken into account, if that makes sense?
Yeah, it's a great question. This is something that comes up also in the gene editing field a lot, right? So one of the I'm going to kind of apply your logic to another question, which I think is very similar. When we do CRISPR-Cas9 gene editing to generate a mutation, there we do clonal selection. And who knows what random mutagenesis occurred from off-target effects of CRISPR-Cas9 editing, of our guides not being specific, et cetera, et cetera. And this is something that a lot of people have kind of tried to bring up in the field. There have been a couple of approaches to hedge against. The first is to first do it multiple times, not only in the same set of iPSCs, but in a different set of iPSCs with a different genetic background. Now, you might get the same genetic manipulations, but if there's some stochasticity in them, you will, at least in the context of lentiviral integration, you might, noise out those types of effects. The approach in the context of, say, a guide and a CRISPR cutting approach, which could have consistent off-target effects on every cell line you look at. One approach that has really been pioneered, and I think by a large genome engineering initiative that I'm a part of here at NIH, the INDIE project, has been to create revertant lines. So what these are is you generate a CRISPR edited mutant of your parental line. Say your parental is a healthy line, you generate the AD risk mutation. And then you take that AD risk mutation line and you reverse the edit with yet another edit. Now, if the phenotype of the parent line and the revertant match, and your AD risk line is not, then the effects that you see are likely not due to the effects of genome editing, but rather effects of off-target genome editing, but rather the intended genome editing. Now, this again has to be verified, and it's always good to do things in more than one line. It's always good to try things more than one time. But this is an approach that I think has really changed the way we think about genome editing in the field. And for those of you who are interested in iPSC lines and neurodegeneration, these are publicly available lines that we're actually releasing through the JAX website. So if anyone's interested, it's a cool strategy. And I think it hopefully helps people do better controlled experiments and avoid these factors.
Very cool. And so for our final question here, we have a few extras. So you primarily mentioned the effect of SNPs on Alzheimer's disease. Are SNPs the most prevalent type of mutation? And what are other types of mutations that people commonly look into?
So there are multiple other types of mutations, like structural variants and other types of changes to DNA. One thing I'd like to make clear is the SNPs that I brought up in the genomic mutation, actually for Alzheimer's disease and late onset Alzheimer's disease, the vast number of them are non-coding. I pointed you to one coding mutation in ApoE, which is by far the strongest and the most probably notorious. But there are a few other coding mutants, but the more common SNPs, by and large, the most common SNPs are non-coding. Sometimes we don't even really know that the gene that's annotated, those Manhattan plots are just attributed to nearest neighbor genes for a lot of the time. Some of them have eQTL analysis to back it up, some of them don't. So we don't already know which gene each SNP affects. Some of them we do, some of them we don't. So there is still a lot of work to even know whether SNPs are in genes. Structural variants, there are structural variants that are associated with other types of neurodegeneration. This isn't something that is as common in the context of neurodegenerative disease, but these issues are emerging a little bit more with long-read sequencing data that's coming out. I think we'll learn more about different types of mutations. SNPs have been the most accessible with short-read sequencing, and I think is, as a result, where we have started.
Very cool. And if you don't mind, one last question, because I think it's a really great question. So in Alzheimer's disease, microglia also play a crucial role. However, gene delivery to them is really challenging. Do you also work on microglia, and how do you deliver genetic cargo to them?
Fantastic question. This is something we've just started to do a little bit of. So I have some practical response to this. So yes, we love microglia. In fact, one of the first studies that has come out of our lab is understanding lipid biology in microglia and how lipid biology can change immune state in microglia. Changing, as you mentioned, if you add a lentivirus by itself to a microglia culture, they are very difficult. And they are naturally immune cells. They react to virus parts. They inflame themselves when treated with virus. So for sure, that is a problem. One way we've tried to get around this, which has been actually working, is use these VLP, these virus-like particles that are supposed to enhance, they're empty, essentially kind of capsids, empty capsids that help, I guess, uptake of lentiviruses. So we've been playing around with these in conjunction with lentiviruses, so co-delivering both of these, and that has greatly enhanced the delivery of genes. And this is something that I think Beth Stevens' lab has reported in a manuscript Nature Immunology not that long ago. And we kind of took after that approach and tried it, and it definitely works. So the thing that I think we're slowly starting to figure out is the timing of that to avoid confounding immunogenicity of these substrates with the actual microglial gene delivery consequences. So we might have to deliver it, wait a bit, and wait till the microglia kind of calm down from being assaulted with virus, and then probe their biology rather than trying to conflate immune biology with other biology that we might be interested in if it's not immune. So, but yes, there are approaches. They are nascent, but the early ones do seem to have some effect, which is exciting.
Very cool. All right, amazing. Well, we are wrapping up just on time. Thank you again, Priyanka, for an amazing talk and really awesome, engaging discussion. It was great to see so many wonderful questions and great to hear so many perfect answers for them. So thank you all of the audience, and thank you so much to Priyanka.
Taking the
I do
time.
want to. I do want to mention that we will have our next edition of the Gene Delivery Seminar Series on October 10th at 11 o'clock Eastern Time with Dr. Kathy Krentz. So please mark your calendars, and we will, of course, be reaching out closer to the time. But for now, thank you all so much again, and I hope you have a wonderful rest of your day. Thanks.
Thank you.